In the year 2000, while eating his lunch in the remote Badlands of Montana, fossil preparator Bob Harmon happened to notice a fragment of fossil bone protruding from the rocky slope above him. That bone turned out to be the femur of a Tyrannosaurus rex, and it was only the first of many fossils uncovered afterward. By the end of the excavation, the team had recovered nearly 40% of the skeleton, including most of the skull, along with several vertebrae and limb bones. This individual was given the nickname Bob Rex, or more simply, B-Rex, in honor of Harmon. The discovery began to fill a major gap in our understanding of dinosaurs, because while we like to think we know quite a lot about them, such as how they moved and what they ate, for a long time, scientists could not determine one seemingly basic aspect of their biology, which dinosaurs were male and which were female. In the case of B-Rex, researchers had to break open part of the fossil and look inside to find the answer. Although paleontologists often give fossils nicknames, we rarely ever know whether to call them he or she, and in this context, male and female are used simply as shorthand for sperm-producing and egg-laying individuals. Being able to identify such traits from fossils is actually crucial, because it allows us to understand dinosaurs as living animals rather than just movie monsters or museum skeletons. It can even reveal insights into their behavior. One way paleontologists have attempted to solve the mystery of dinosaur sex is by looking for repeated patterns across skeletons of the same species to see if they can be divided into two groups, one group showing a particular feature and the other lacking it. This is because many of the dinosaurs' closest living relatives are sexually dimorphic, meaning males and females differ noticeably in their physical appearance. Take size, for example. In some species, there are significant differences between the sexes, with males often being larger than females, usually so they can directly compete with one another for mates. Or consider the extravagant feathers of male peafowl, which they display to attract the attention of their more plainly colored female counterparts. But sexual dimorphism does not always mean larger or more brightly colored males. In red-eared slider turtles, females are actually bigger, their bodies enlarged to hold more eggs. And in spiny-footed lizards, it is the adult females that have vivid red tails, a feature the males seem to prefer when choosing mates. This shows that a trait signaling maleness in one species can instead signal femaleness in another, and identifying the sex of an animal can tell us a great deal about its behavior and reproductive strategies. In the 1990s, some paleontologists proposed that female T. rexes might be identifiable by the shape of their pelvis, and that they could have been the larger sex, similar to red-eared slider turtles. But they had no firm evidence. For living animals, the quickest way to determine sex is through their reproductive organs. Males have testes, and females have ovaries. Dinosaurs, however, did not preserve such organs. Soft tissues like skin, muscle, and internal organs typically decay long before they have any chance to fossilize, making them extremely rare in the fossil record. So far, the closest evidence of a fossilized reproductive organ is a remarkably preserved cloaca from a Psittacosaurus, the single opening used both for reproduction and for expelling waste. But even this extraordinary fossil did not preserve the internal structures, like testes or ovaries, that would allow scientists to determine whether it was male or female. It is possible that some dinosaur species truly were sexually dimorphic, since differences among individuals have been observed. Yet, without being able to definitively identify the sex of any single specimen, it is difficult to say whether these traits were truly linked to sex at all. The differences could just as easily reflect sexual dimorphism, age-related changes, or simply natural variation within the species. In some cases, they might even mean that two entirely different species were being confused. What paleontologists really needed was an independent line of evidence, one that could directly link a skeleton to its sex, rather than relying on interpretation alone. And so the question arose: what if the difference between male and female dinosaurs could be found deeper within the bones themselves? In 2002, when B-Rex was being excavated, the team faced a particular challenge. The site was located in rugged terrain with no roads, which meant the fossils had to be transported out by helicopter. The largest block, containing B-Rex's legs, weighed more than 900 kilograms, beyond the helicopter's weight limit. As a result, the team had to carefully divide the block into two sections. During this process, one of B. Rex's femurs broke apart, splitting into several fragments. A broken dinosaur bone might sound disastrous, but for scientists who study the internal structure of fossils, it was a rare opportunity. Paleontologist Mary Schweitzer, who had devoted her career to investigating the complex structures hidden within fossilized bones, examined the fragments under a microscope. There she identified a special kind of tissue that could help solve the question of sex, at least for this individual. What she saw was a tissue known as medullary bone, a spongy material found only inside the bones of modern female birds when they are preparing to lay eggs. This bone partly fills the hollow cavities in bird bones, which make them light enough for flight, and it serves as a ready reservoir of calcium for building eggshells. Schweitzer had long studied bird biology to better understand their connection to their non-avian dinosaur ancestors, so she immediately recognized its structure. To confirm that what she observed in B. Rex's femur was indeed medullary bone, Schweitzer compared it with bones from ostriches and emus that had died during their egg-laying cycles. The porous, irregular texture of those samples was nearly identical to what she found in B. Rex. This was an extraordinary discovery. It seemed like a near-certain way to identify the sex of a dinosaur, at least in this case. If Schweitzer was correct, then Bob Rex was in fact a female. And not just a female, but one that had been gravid, carrying eggs at the time of death. While many had long suspected that theropods, such as T. Rex, might produce medullary bone like their avian descendants, this was the first direct evidence. Yet the debate was far from over. In 2009, another research team described unusual bone tissue from a diseased turkey vulture and from a Cretaceous dinosaur found in Transylvania. They noted striking similarities between these samples and those from B. Rex. They suggested that what Schweitzer had identified might actually have been the result of a bone disease known as osteopetrosis, which affects many modern birds. They also argued that giant dinosaurs like T. Rex may not have needed medullary bone at all. Medullary bone in birds is thought to have evolved as a safeguard against their thin-walled, fragile bones, which could easily fracture if calcium were withdrawn directly. By contrast, medullary bone provided a safe supply of calcium for eggshell formation. But in animals with thick, dense bones, like crocodiles, calcium can be drawn directly from the skeleton without risk. And since the bones of T. Rex were also relatively thick, the researchers concluded that these dinosaurs may never have needed medullary bone in the first place. By 2014, a new study reported evidence of medullary bone-like tissue in the lower jaws of several pterosaurs. But pterosaurs are thought to have laid eggs with thin, leathery shells that required little calcium. This raised a new challenge. Perhaps medullary bone was not solely related to reproduction. At the time, it was also believed that medullary bone formed only in limb bones. So if it appeared in the jaws of pterosaurs, it might have had another function beyond egg-laying. And if that were the case, it could no longer be trusted as a reliable marker of a gravid female. The debate therefore continued. Did B. Rex truly preserve medullary bone? And do we have any certain way to recognize female dinosaurs? To strengthen the case that B. Rex's medullary bone was genuine, Schweitzer and her team carried on with their work. In 2016, they published a study analyzing the B. Rex bone at the molecular level, directly comparing it to the chemical signature of medullary bone in birds. They applied special stains that react with molecules unique to medullary bone. The B. Rex sample did react, turning dark, brown, and blue, just like the samples taken from a hen and an ostrich. They also tested the stains on bone samples from chickens with osteopetrosis, and those did not react. This suggested that if B. Rex's bone had only been diseased, it would not have reacted either. In 2019, Schweitzer collaborated on another study showing that birds in their egg-laying cycle can form medullary bone throughout much of their skeleton, even in the lower jaw. This meant that the reproductive role of medullary bone could not be ruled out simply because it appeared in unusual locations. So, where does the debate stand today? These analyses persuaded many scientists that medullary bone can indeed be useful for identifying mature female dinosaurs. But some paleontologists remain unconvinced. Recently, a study of 61 ornithomimosaurs that died together in a single herd revealed differences consistent with sexual dimorphism. This indicates that there are still alternative approaches to determining dinosaur sex, provided the right samples are available. All of this shows that understanding dinosaur biology is more complicated than it may seem, even for a species as famous as T-Rex. Yet with every hypothesis tested, we move closer to reconstructing what their lives were really like. Thank you for watching today's episode. If you enjoyed this content, make sure to like the video and subscribe to the channel.